Inside your eye, light doesn't just illuminate the world, it transforms into signals your brain can interpret. This process, called phototransduction, happens faster than you can blink, about 13 milliseconds. Your eyes work tirelessly, capturing light and translating it into the rich tapestry of images you see every day. Yet light itself is nothing more than electromagnetic waves. How does your body make sense of it? The secret lies in the retina, a thin layer of cells at the back of your eye. These cells detect light and initiate a cascade of reactions, converting waves into neural messages. This is the first step in a complex visual journey. The retina houses over 100 million photoreceptor cells, each specialized to detect different light intensities. Rod cells let you see in low light, while cone cells enable color vision. Amazingly, humans can distinguish over 10 million shades of color. These cells form a delicate mosaic, each reporting back to the brain through the optic nerve. But how do they activate this neural relay? Rods and cones contain pigments that react to light. This reaction shifts the structure of the pigment and sets off a chain of chemical events. It's like flipping a switch that illuminates an intricate network of signals. Your eye's ability to focus and capture sharp images begins with the cornea and lens. Together, they bend incoming light, directing it precisely onto the retina. The lens adjusts its shape to focus on objects at varying distances. This dynamic change is known as accommodation, a quick and seamless transition. The retina's surface is curved, allowing it to project the image upside down. But why upside down? It's a simple optical consequence of the lens's curvature. Your brain, adept at interpreting these signals, flips them right side up without you even realizing. A perfect example of your body's invisible efficiency. Light-sensitive cells in your retina are not evenly distributed. The center, the fovea, is packed with cone cells, allowing for detailed, color-rich vision. In contrast, the periphery contains more rod cells, enhancing night vision. This distribution is no accident, it optimizes both day and night functions. But there's a catch: the optic nerve exit creates a blind spot. Surprisingly, you don't notice it. Your brain fills in this gap, using surrounding information to create a seamless view. This compensatory mechanism highlights the brain's remarkable capability to adapt and interpret incomplete data. At the intersection of light and perception lies the optic nerve, a crucial conduit. Composed of over 1 million nerve fibers, it serves as the highway for electrical signals. Each signal represents a fragment of the visual scene your eye has captured. But these are not mere impulses, they carry encoded data about light intensity, color, and contrast. The optic nerve transmits this information to the brain's visual cortex, where images are reconstructed. This entire journey from light capture to brain interpretation occurs in milliseconds. Every blink, every glance is a testament to this ceaseless, intricate ballet of light and neural processing. Deep within your retina, photoreceptor cells contain stacks of membranes loaded with light-sensitive proteins called opsins. When light hits these proteins, they undergo a shape change, initiating a biochemical cascade. This cascade involves a molecule called retinal, which switches from a bent to a straight form. This tiny structural change amplifies the signal, triggering the breakdown of cyclic guanosine monophosphate (cGMP). The drop in cGMP levels causes ion channels in the cell membrane to close. As these channels close, the cell's electrical charge changes, producing an electrical signal. This is the birth of a visual signal, a remarkable conversion of light into an electrochemical impulse. Once the photoreceptor cells generate electrical signals, these messages travel across the retina through a network of neurons. Bipolar cells receive signals from rods and cones, relaying the information to ganglion cells. Here, signals converge and are refined, highlighting edges, motion, and contrast. This preprocessing sharpens your perception of the world. Astonishingly, ganglion cells are responsible for transmitting over 1 million signals to the brain every second. They encode information into action potentials, brief changes in electrical potential that race along the optic nerve. This neural choreography ensures that your visual system operates with unparalleled efficiency. Inside the ganglion cells, advanced circuitry processes and refines visual data. These cells are equipped with specialized ion channels that respond to neurotransmitters, adjusting their firing rate. Horizontal and amacrine cells contribute to this intricate network, enhancing contrast and motion detection. For example, amacrine cells help detect movement by comparing signals between neighboring cells. This layered processing allows your eyes to differentiate between static and moving objects. Such precision enables you to spot predators, prey, or moving vehicles even at high speeds. The entire system is a testament to nature's genius in combining simplicity with complexity. Your brain's visual cortex is where the final stage of image processing occurs. Here, incoming signals from the retina are decoded and interpreted. The visual cortex is divided into specialized areas, each responsible for processing different aspects of vision, such as color, shape, and motion. Neurons in these areas form specific pathways, creating a detailed map of the visual field. This mapping allows you to recognize faces, read text, and perceive depth. The cortex's ability to integrate diverse signals into a coherent image is a pinnacle of neural processing, completed in a fraction of a second. This seamless integration allows you to navigate and interact with your environment effectively. The entire visual processing pathway relies on a delicate balance of chemical and electrical signals. Neurotransmitters like glutamate play a key role in transmitting signals between cells. But the system is not just about speed, it's also about adaptability. Your visual system continuously adjusts to changes in light intensity and focus, a process known as adaptation. This allows you to transition from a dark room to bright sunlight without being overwhelmed. Feedback loops within the retina and brain ensure that your visual perception remains stable despite varying conditions. This adaptability ensures that your vision remains reliable, no matter the challenges. When your visual system faces extreme conditions, it showcases its resilience and limitations. In low light, rod cells dominate, allowing you to see in the dark. Yet this comes at a cost, a lack of color perception. Your eyes need about 20 to 30 minutes to fully adapt to darkness, an adjustment called dark adaptation. In contrast, bright light temporarily overwhelms photoreceptors, causing them to desensitize in a process known as bleaching. This is why stepping into sunlight feels blinding initially. These adaptations highlight the eye's ability to withstand drastic changes, ensuring that you can function in varied environments. Your eyes continually balance sensitivity with accuracy. Sometimes, the visual system can be tricked or reach its limits. Optical illusions exploit the brain's assumptions, revealing how your visual processing can be fooled. The Mueller-Lyer illusion, for example, makes identical lines appear different in length due to surrounding angles. This occurs because your brain uses context and past experiences to interpret visual input. Another limit is color blindness, affecting around 1 in 12 men and 1 in 200 women worldwide. It results from genetic variations affecting cone cell pigments. These conditions expose the fine balance between perception and reality, showcasing how the eye and brain collaborate to interpret the world. Under stress or fatigue, your vision might blur or become less reliable. Eye strain from prolonged screen use can cause discomfort and temporary blurriness. This is due to reduced blinking, which dries out the eyes. Prolonged stress can affect blood flow, impacting how nutrients and oxygen reach your eyes. This can lead to temporary vision changes like seeing spots or flashes of light. Migraines can trigger auras, visual disturbances that precede headaches. These phenomena underscore the sensitivity of your visual system to internal and external pressures, emphasizing its need for regular care and rest to maintain optimal function. Rare conditions offer insights into the visual system's intricacies. Synesthesia, where stimulation of one sense leads to automatic experiences in another, can cause some to see sounds as colors. This condition reveals the interconnectedness and plasticity of sensory pathways. Another condition, aphakia, results from the absence of the lens, allowing ultraviolet light perception. These anomalies demonstrate the flexibility and adaptability of human vision. They highlight how even minor changes in structure or function can lead to profound differences in perception. Such cases provide unique windows into the brain's interpretative processes, expanding our understanding of sensory integration. The process of converting light into images is astonishingly precise. Your eyes can detect a single photon, the smallest unit of light, highlighting their incredible sensitivity. This means that in absolute darkness, if just one photon strikes a rod cell, it can trigger a visual response. This capability underscores a fundamental truth: your eyes are not just passive receivers but active interpreters. They filter, prioritize, and convey information with unerring precision. This single fact reframes our understanding of vision. It's not just about seeing, but about the profound interplay between light and biology, a dance millions of years in the making.